Hello, thank you so much for tuning into my YouTube channel. In this video, I want to talk about the fact if everyone sees the same color the same way. This is a color perception problem, and there has been quite a few papers in this area. One of them in particular has been published by Asano and his colleagues. And one of his colleagues is actually my master's advisor, and we're gonna be focused on that paper. And I've also done some work in this area, which I'm gonna explain more in the video. Let's get started. Okay, as I said in this video, I want to talk about the fact if everyone see the same color the same way. Color vision is not the same from person to person, meaning different people do not see the same thing in the same way. And it is not only limited to color deficiency, but also a huge variability that is present among individuals with normal color vision. A set of three spectral functions, which are known as color matching functions, short for CMFs, which would be shortened to CMFs, would be able to characterize the human color vision. These three functions are linearly related to the spectral sensitivity of human observers. The spectral sensitivities belong to three cones responding in a different way to light of different wavelengths. These cones are represented by L, M, and S, showing their peak sensitivities in long, middle, and short wavelength regions, respectively. So these are CMFs, color matching functions, and these are the sensitivity of human observer. These two are linearly related to each other. And these are the cones. The red represents the long, and blue represents the short, and green represents the middle wavelength cone that are sensitive to long, middle, and short wavelengths. As you can see, people, they don't see the same color the same, and there is variability. Therefore, human color vision variability, in a sense, would equate to the CMF's variability, which equates to the spectral sensitivity variability between people. So people, they don't see the same color the same. That means their color matching functions are not the same. Which means their spectral sensitivity is not the same. They're all related to each other, and it's very customary that people don't see the same color the same. Not even two people. Even two people, they don't see the same color the same. CMFs consist of lens, macular pigment, and three kinds of photopigments, namely L, M, and S cones. Each constituent possesses inter-observer variability, contributing to the total CMF's variability. So lens, macula. Macula is a yellow filter that protects fovea from light of short wavelength, and also it helps with the chromatic aberration that happens because of the short wavelength not being focused through absorbing the short wavelength. So lens and macular, they both are very effective in our CMF's variability, which will lead to us seeing the same color, not the same. Even though there is a significant variability between observers when it comes to CMFs, the majority of the suggested models up until now for observers are average observer functions, like CIE 1931 standard observer and CIE 1964 standard observer. These are the two. I showed one of them in the first slide. The 1931 is 2 degree and 1964 is 10 degree visual angle. So these are just average observer. CIE PO 06 was the only model that considered a change in age and field size in terms of visual angle and in the resulting CMFs. There have been a small number of research trying to consider the individual's variability in terms of CMFs using Monte Carlo simulation and predicting a color normal population. CMFs was first proposed by Fairchild. So Fairchild was the one who came up with the use of Monte Carlo. He was also my master's advisor. Very knowledgeable man. And the model was then enlarged on by Fairchild and Hackman through taking 4 physiological parameters, namely lens pigment density, macular pigment peak optical density, the maximum wavelength shifts of L, M, and S cones as inputs. The most recent research in this area belongs to Asano and his colleagues. They came up with a model in which there were 10 parameters as inputs in order to more precisely model individual CMF's variability. 2 of the inputs were age and visual angle. The other 8 inputs were physiological parameters, which include deviation from average for lens pigment density, deviation from average for macular pigment peak optical density, deviations from average for L, M, and S cone photopigment peak optical densities, and finally deviations from average for L, M, and S cone photopigment maximum wavelength shifts. The suggested model by them in order to predict the color normal populations, CMFs made use of Monte Carlo simulation. In order to be able to perform the Monte Carlo simulation, a probability distribution is needed. They made an assumption that each physiological parameter would form a normal distribution around the mean. Therefore, in 2 stages, the standard deviation of the physiological parameters were specified. At the first stage, past studies were investigated thoroughly, and their reported standard deviations were collected, averaged, and then used in the work. In the second stage, the standard deviations attained at the first stage were scaled to fit the target color matching dataset variability. To be more concise about the model they propose and make it more understandable, CIEP-006 would set the baseline CMFs utilizing the supplied age and field size data. The proposed model by Asano and his colleagues would deviate the baseline CMFs in a random fashion using the eighth additional physiological parameters. The model proposed by Asano and his colleagues has been shown here, as you can see, where A is the age of the subject, V denotes the visual angle, D lens mark the deviation from average for lens pigment density, and D macula shows the deviation from average for macular pigment optical density. DL, DM, DS, they denote the deviation from average for L, M, and S cone peak optical density respectively, and SL, SM, SS, they show deviations from average for LM and S cone maximum wavelength shift. Consequently, the basic input forming the baseline, which are inherited from CIEP-06 to this equation, are A and V, age and visual angle. Now let us examine them more precisely, more closely, and see how they would affect the resultant CMFs of a specific observer when changed using Monte Carlo simulation. So I'm going to just use the model proposed by Asano and his colleagues, and I'm going to just and I'm just going to show you guys how A and V are going to affect the output LMS CMFs. This figure shows the CMFs of a subject age 20, but using 2 and 10. But the size of the field, the visual angle is not the same. The first one is 2 degree and the second one is 10 degree. So the the size of visual angle is not the same. And as you can see, field size can alter the CMFs obtained in an almost significant manner. Let us now have a look at age. Using the 2-degree field size, I'm going to simulate the observer whose age would alter from 20 to 55. In other words, we want to see how the increase in age would affect the CMFs. This is the CMFs varied from 20 to 55. Now that we have some idea about the baseline of the model devised by Asano, we proceed to introduce the physiological parameter used by him in order to deviate this baseline. Okay, let's go quickly over the equations they used. They first obtained the lens and other ocular media spectral optical density. Then they obtained the macular media the macular pigment optical density in terms of spectra, and then they obtained photoreceptors, absorbance spectra, and after that all these things should be multiplied by one another, because they're sort of like filters on top of one another, so the light would go through them and the spectra would be reduced depending on the absorbance of the media that the light is going through. So they all multiply by each other, and it leads to cone fundamental in terms of quanta. Cone fundamental means the spectral sensitivity of the cones, but also considering the macular and ocular media effect on the light. And then at the end, the cone fundamental in terms of energy would be obtained through multiplying them through multiplying these equations by wavelengths. So this is the equation that they used in the Monte Carlo in their equations, and they also use Monte Carlo simulation to simulate observers LMS CMFs. We could say observers CMFs that would be simulated using Monte Carlo. So in this work, I also imparted another variable which would consider the effect of smoking. Smoking would affect the lens optical density. Basically, the same model as Asano's would be used, but some modifications are made to it, which considers the smoking status of the observer. So this is the study that they done. They came up with equations showing the relationship between lens optical density and the age of the smoker and non-smokers. Here are the equations, where x shows the age and y shows the lens optical density. And this, as you can see, the slope of the smoker is twice as much as the non-smoker. So we use this one, and we multiply the ocular media ocular media optical density. We multiply by 2 if the observer is a smoker. And if the observer is not a smoker, we just leave it the way it is. So the smoking is it would reduce the amount of light that would go through the lens. So one of the suggestions is just stop smoking, but it would be interesting to see what happens. So just as a repetition again, I just want to repeat myself again that so the ocular media density. Ocular media optical density would be multiplied by 2 depending on if the observer is a smoker or not. So here are the CMFs belonging to a smoker observer compared to a standard CMFs belonging to 1931 standard observer. As you can see, there is a big shift if someone smokes for the smoker observer. And if we really want to have a good color reproduction device for everyone, we might have to consider all this variability between the observers. There is still debate also going on whether or not we should consider the individual variation, or we are fine doing what we've been doing using the average observer, which has also been shown here, as you can see. So we're still using the average observer, standard observer, but you can see that there's a significant variability between these two. Now, to have a better understanding of how these two see the same thing, I'm gonna visualize the Macbeth color checker, the color chart that I've been using in my videos. I'm gonna use that and see how this color chart would look to a standard observer as opposed to an observer who smokes. So this is the standard observer and this is the smoker. As you can see, the red is more reddish. As you can see, there is a difference between these two rendered Macbeth color checker. One seen by a standard observer on the left, and the other seen by a smoker normal observer on the right. You can see that the blue is not as saturated in the case of the smoker, and red is very red, and orange has also become stranger. If you look at the orange here, if you look at the blues here, on the left, blues are more blue or bluer, and the red on the right is redder. While yellow has also become more reddish, which makes sense. If you look at the yellow, it's more reddish compared to here, compared to the left. So this makes sense, as when we smoke, the lens becomes yellower, leading to the yellow. The lens becomes yellower, leading to the blue light becoming more scattered and absorbed, which results in the red becoming stronger and blue becoming more scattered. So that's why on the right side, which is a smoker, the blue is less blue, but the red is more red. The yellow has also been pushed towards red, and orange is also red. Everything looks more red because the observer lens scatters more blue compared to red. I think that's about it. Thank you so much for watching this video. I hope you liked it and you've learned something useful. I personally really enjoy this topic, and it never ceases to amaze me to see how as people we're not the same even in color vision. And if you like the video, don't forget to subscribe and share it with your friends. Thank you so much and have a nice day.